I could finally say that we have everything, every person at the right place to finish what is to achieve what is our mission.
Okay, hello everyone. Thank you for coming and taking the time to join our conference call for this quarter. Hey Enzo, how's it going?
Hi, Ryan. Very well, thank you. How are you?
Yeah, not too bad. I know we're in very different time zones, but it's always good to connect. We have a lot of exciting things to share. A lot has happened since the last time we had a conference call. To start with, as you know, the animal studies are finished. The reports for both the rat trials and the sheep trials have been written and are in the process of being submitted to journals for publication. So they've been submitted to journals, and now both of them are going through the process of getting published. We've also commissioned a new histology study with Dr. José Vega. As you'll see later in the call, he's a specialist with over 40 years of experience. He is looking at the nerve structures of the foreskin. Enzo did a great job in finding Dr. Vega and building that relationship and organizing that study. Also, what's very exciting is we started a new partnership with Dr. Stanislav Žiaran in Bratislava, Slovakia. It's a comprehensive research project. The purpose of it is to complete all the remaining preclinical objectives, so to get all the data that we need to be ready for the human clinical trials. And we'll go over that a little in more detail during this call. And then lastly, we also had a successful GoFundMe fundraising campaign. So thank you to all of you who contributed in that fundraiser. Also, I know you contribute monthly as well, and it's extremely generous of you, and we are very appreciative of the support you give us because you are making this research possible. All of these contracts we have, all these projects we have going on, it's all made possible by you. So we're extremely grateful for that. So give yourselves a pat on the back, and we're doing we're making the most effective progress we can with the donations that you give. So let's move into a PowerPoint presentation. We're going to briefly give a presentation on some of these research updates and what our plans are for the near future. Now, Enzo, I'm going to turn it over to you. If you'd like to introduce some of our new research partners to the donors and talk a little bit about who they are and what their expertise is and
Yeah,
what they're doing for us.
basically, we at a certain point during the previous research with Italian scientists, we wanted to look for other possible options for decellularizing and repopulizing tissue. And so, this collaboration starts from this specific need. And I found these scientists in Bratislava, wonderful guys, very prepared, very humble guys for what they really know or what they really do. And, they are specialized. So the good thing is that they are doctors, urologists, and at the same time, they are biotechnologists, so, tissue engineers. And so, we have finally in one person these different disciplines. Dr. Ziaran is the guy I spoke at the beginning, and then he introduced to us his PhD, Jan Kovac. And as the donors might know, we did this wonderful trip to Bratislava to meet them personally. And, it was a very fruitful trip because we could talk each other and feeling our own emotions. It's much better to talk watching in the eyes. And we realized these are very good people, first of all, which is very, very important for our ethical work, and very, very prepared scientists. Also, this is what I have to say about them.
Yeah, thank you, Enzo. Dr. Ziaran, his assistant, Jan Kovac, is a bioengineer with experience in the 3D bioprinting and also bioreactors, which is going to be very critical for us. William, perhaps you'd like to fill in some of the technical details on their work with bioreactors that's applicable to our project?
Yeah, absolutely. Can you hear me okay, Ryan?
Oh yeah, you're loud and clear.
Perfect. So yeah, one of the big additions is there's the use of bioreactors. So in this case, in the research project that they're working on, in one of the milestones, we're actually working on recellularization inside the bioreactor. And they're taking a lot of time and care in the design of that experimentation. So, like they were very concerned about microplastics and stuff like that. So they actually gave us an update recently and showed us some of the techniques they were using in the bioreactor. So that's still underway. They're still working on that. They're still developing that procedure. In fact, I think Ryan, we just reviewed one of the protocols last night, um, or my nighttime.
Yeah, they're already producing a lot of data that they send to us, and we give feedback guiding the project so that it suits our needs. They're very productive. They're very eager to work with us, and they hit the ground running. So we're very excited to work with them. Next, we have Dr. José Vega at the University of Oviedo in Spain. Again, Enzo, would you like to fill us in a bit about Dr. Vega and his background?
Yeah, Dr. Vega is the dean of the Faculty of Medicine of the University of Oviedo in Spain, and he's basically the most expert person I have ever known about um, nerve endings of the foreskin in the world. So he has more than 45 years of experience in this specific topic. And when I went to meet him personally in Oviedo last summer, he explained me very, very, very important details that make the difference, especially because he didn't know what we were doing, and we didn't know what he was doing. And when we put together these two knowledges, we understood we could do a lot of work together. Nerve regeneration is something that it's already been described very well in the scientific literature. But adding a person like Dr. Vega, who is definitely a great expert of this field, makes our project more, gives a lot of authority, I would say, which is what is important, I mean, for us. So I'm very excited to work with this new scientist, Spanish and Slovakian guys.
Absolutely. And as we complete more research, so Dr. Ziran's team, they're working on getting all the data together so we can make a package at the end that we use when we go to apply for ethics approval to conduct the human clinicals. We send data from our other partners like Dr. Vega to them, and they include that in their project. They've received data from Histologics. They reviewed the animal trials results, or filled them in on that from Italy. And so they're incorporating all this data into their project so that at the end we can have a nice wrapped up uh, portfolio that we can use when approaching the regulatory authorities. So, as we've recently, last year we hired Beatrix, who's our clinical project manager, and she, among the things that she brings to the table, is her experience in preparing for and administering human clinical trials. So she always reiterates that it's very important to be thorough and comprehensive, and it's almost like you can never have too much data. So we want to get as much data as we can together before we go to approve or apply for human trials. And Dr. Vega is a part of that, a very important part of that. This is the overall research project with the different tracks that we have going on. So it's a rough bar chart, but basically, if you look from 0 to 10, that's how far along are we to completing a particular research track. You can see with the animal studies, we're at a 9 because they're finished. The reports have been written. They're just in the process of being published right now. With the histology studies, we're basically at a 7. Histologics submitted their first draft report to us that is currently under review, and we're deciding how to move forward with that. And then Dr. Vega's study is almost complete. So I think roughly within 1 or 2 months that should be finished and nearing publication, but we'll see. It's always hard to get a solid picture on timelines with publishing studies. And then with the preclinical project over here, which is what Dr. Žiaran is working on, as you probably saw in the recent YouTube presentation we put out a few days ago, there are 4 primary milestones for that project, and the first 3 are being done in parallel. And we had a call on Friday with the team, and they gave us an update on their status. So as you know, the goal is to harvest 15 to 20 tissue samples and then move every one of them through the milestone. So through the decellularization process, the recellularization process, and then running them through the bioreactor to perfect the protocols for each one of those stages. They've harvested 10 samples and they've already decellularized 4. So they are, in the swing of things and moving through the project. And like William said, that we on the call that we had with them on Friday, they shared the data they have so far on the decellularization protocol. So William and I reviewed that, and we're going to do more internal review and then get feedback back to them. So that project is in full swing, and we're very excited about the progress that they've achieved so far. And as stated previously, we estimate that project will be finished by the 4th quarter of this year. So that's our current estimate. Then with human trials, we have 1 block here finished, basically because we are actively in the process of completing the preclinical project. And once that's finished, we will, once that project is finished, the next steps are to finish the surgical plan development, which should be done pretty quickly. And then we have to identify a suitable CRO to partner with and decide where we want to do human clinical trials and then apply for ethics approval. So, of course, we will keep you updated at every step of the way. William, would you like to share some, like, a technical explanation of each of these milestones and how they're functioning and what they intend to accomplish?
Yeah, absolutely. Milestone 1, it seems pretty easy and self-explanatory with the donor collection. That was actually one of the big items that we were talking about the other day. We have to make sure that that is a vetted, and now is the time for us to go through and make sure everything's working, make sure that we have the correct procedure for doing this. And again, we have a really good team. We finally found our A team, so to speak. Dr. Žiaran is an MD PhD. He's a urologist, so we're using him pretty heavily for input on protocols and developing protocols. So that was actually something we talked about yesterday, going through their protocols for collection. So this is from cadavers, and this is actually at the hospital that Dr. Žiaran works at. Milestone 2, dynamic decellularization, and then dynamic recellularization. And if you'll notice, 1, 2, and 3 are all kind of happening at the same time. This is one of the first times that we've been able to do that, is really fast track things, get things moving faster. And I think that speaks to how awesome and professional this team in Bratislava is that they were eager to help us with that and get things moving faster. So the dynamic decellularization is they're working on the best way, like what is the best method? Because they have a couple different methods of doing it, and they're trying to figure out what the best method is. So the process in like a high level broad spectrum is they're doing a decellularization process that they already have, or that they've already documented works, and then they're sending the foreskin tissue out for histology studies in their own lab. So they, within their group, they have some researchers that do histology pathology. So they're making sure that they've done a clean decellularization. They're making sure that it meets all the criteria, that there's as minimal possibility for rejection as possible. but also that all the proteins and all the stuff that we need to recellularize are there. So that comes into the characterized decellularization, is it hitting the targets that we want removed? Is it leaving the things we want to stay? And then minimize variability, obviously. We want it to be the same every time. We don't want any surprises. And then that goes into the dynamic recellularization, which is just like decellularization, we're trying to optimize the recellularization. So they're working on the best method for that. And this milestone is kind of interchangeable with milestone 4, which is using the bioreactor. Because if we need to pivot at any time and say, you know what, we just need to move over to the bioreactor, we have the ability to pivot. If we're not set in stone, it eliminates hurdles for us. So one of the biggest things we're trying to avoid is a stop. Like, we're moving forward, we're getting research done, and now there's a problem. We have to stop and think about the problem. We already kind of have a backup built into this where we know we're gonna do the bioreactor. If we need to do the bioreactor sooner, they're already developing that and already working. They already have a working bioreactor model. The cool thing about the bioreactor is that it's going to eliminate a lot of the guesswork with the body recellularizing. It's going to be recellularized before it goes into your body. So protocols for implementing the tissue and fully characterizing the tissue. So after we recellularize a few in the bioreactor, we're actually going to send those out to histology to see how closely they can match the natural foreskin. So that's a big step that we're coming up on, that once we get a couple of successful, quote unquote, recellularized tissues from the bioreactor process, we're going to, again, within the internal Bratislava team, they have some people that do the histology and pathology. So we're going to send it to their professionals. So it's all in the same location. We don't have to send it over 3 countries away. Hopefully a very quick turnaround time on that. So milestones 1, 2, and 3. And again, 2 and 3, the dynamic decellularization and recellularization are really key components to what we're trying to do. Obviously, I mean, we've been talking about decellularization from day 1, but the bioreactor in milestone 4, again, it's a separate milestone, but it is if we need to pivot, if the team finds that the bioreactor is going to be the dynamic decellularization key method, or sorry, recellularization, then that's where they can immediately pivot to that. But the fact that we can move forward on several different fronts at the same time is a huge plus for us with this team.
Yes, and William, we had discussed this previously, but if we do choose to pursue the bioreactor option, this could also be an alternative to the 2-stage burial method that we had explored in Italy. But since we already did that experiment in Italy and have the data, that could be an alternative. So now, let's say we pursue the bioreactor and we have good results, but for some reason we're unable to pursue that method when it comes to applying for ethics approval, and they say they won't approve that, then we could revert to the 2-stage method and vice versa. So this kind of redundancy is there to save time in the future in case we do run into an issue with one or the other.
Yeah, that's a really good point, is that um, we're really trying to set this up to have a successful path, even if we do get a no in any one of the regards from a governing body. So we have the option to go with a purely surgical method for recellularization, and then we have options. And the bioreactor method also kind of solves. We're hoping that will help us solve another issue of limited sample ability, that which is another project for after we get through approval with this team, looking at how we can maximize using the bioreactor to create more samples. As we all know, that there's only so many cadavers that are donors, and that's kind of a choke point for once this gets approved. So yeah, that is another really good point there too, Ryan.
Yeah, this team does have experience working with 3D bioprinting. So although we're not working with them now on that, and we haven't made a decision on that, that is a potential in the future. If we were to look into bioprinting to resolve the tissue supply problem after human clinical trials. So thank you, Enzo and William, so much for your input on this presentation. I mean, is there anything Enzo or William, you'd like to add to our presentation before we give it to the donors for the Q&A?
Well, what I want to say that just because I have seen all the, since when the company was founded, and I could finally say that we have everything, every person at the right place to finish what is to achieve what is our mission. So we are definitely in a very good position, and we are working with very authoritative scientists that know very well what they are doing, and I'm very happy for this because I could see since the beginning, since 2010, when I founded the company, the development and the milestones and the achievements that we have achieved.
Ryan, to add to actually something Enzo mentioned about Dr. Vega is one of the things that Dr. Vega is definitely very concerned about and offering his expertise in helping us try to solve is the recellularization of nerve endings, mesner corpuscles, and the other cell lines that are involved in light sensory reception. And Dr. Vega has a lot of expertise in this field, and he's working on essentially different protein pathways and different cytokines to help solve that. It's not an immediate fix, but it's something that after the procedure might be a viable treatment to regenerate some of those nerve endings or entice nerve endings to grow into the new tissues. So that's definitely something we're thinking about. It right now it's kind of in the research hypothesis stage with Dr. Vega. He's got a lot of ideas, and once we get closer to the human clinical trials, some of those will hopefully we'll have a lot more information to share on those at that point. I just wanted to throw that out there that we're definitely still concerned about that. We're definitely still thinking about those issues.
Yeah, William, that's great that you bring that up, and I actually wanted to kind of throw it back to you and give you an opportunity to add some clarity on the topic of reinnervation. It's this question comes up often, understandably. That's a very, very important aspect of this project, and there is some confusion, I think, sometimes about how nerve regeneration works and how possible it is. And I think maybe you can talk a little bit about general reinnervation and how it works and how it's been proven in many other projects that it is possible. Do you think you could just give a basic overview of how it works, why it's possible?
Yeah, absolutely. So the idea right now, the standing hypothesis of what method will work best right now is that the like cytokines are a type of protein in the body that entice a cell to move around. So if you get injected with a certain type of cytokine, let's say to move chondrocytes, bone cells, right? Um, when you have that cytokine in a certain location, as that starts to circulate in the body, it entices those cells to move and migrate to the in some cases toward that location where that protein is heavily concentrated. So an injection of a cytokine that entices the movement and the migration of like mesner corpuscles per se might might be a viable option. Again, this is still in the uh, review stage. So Dr. Vega has a couple ideas of a few different cytokines that he wants to use and a few other chemicals actually. But that's still he's still discussing that uh, with the Dr. Žiaran and his team as well. So that's something still in the works. We're not exactly sure which chemicals, but that's just the gist, the overall mechanism that we're thinking will probably work best for us right now. And that would entice your body's own cells to grow into that new tissue and create new nerve endings there.
I wanted to add something to your very good explanation. Because I receive sometimes some email from donors who ask me this specific topic. nerve regeneration doesn't occur immediately after the transplant. It's going to take months because the growth factors allow the nerve to re-innervate the foreskin in a long period. I know from after having spoken this with Dr. Vega that the distance that it's going to take like it's going to make 1 millimeter a day. Okay, so the nerve regrowth. And this regrowth, obviously, even if it's let's say in the dorsal nerve of the penis, it doesn't start from there. It has to start from the spinal cord. That's why it's going to take some months. But this is, it's we know that in every scientific publication about reinnervation, this is pretty clear. I just want to say this because it's going to take months, but in a few months we hope to achieve full nerve regeneration after the transplant, after the surgery.
Great. Thank you both. So at this point, we can move into the Q&A.
Disclaimer: the questions in this segment came from our donors during the conference call, either through chat or by speaking directly. To protect their privacy, each question has been transcribed and then recorded using a generic voice. Question number 1. Why does the regrowth need to start from the spinal cord rather than the dorsal nerve?
Well, I think that to answer to your question, the regrowth starts from the penis because there are nerves in the penis, but the impulse starts from the spinal cords. It's if I can say, it's like that in the spinal cord there is a kind of a small part of the brain, if I could say like that, in a certain way that moves the nerves from of the legs and the sexual part of the persons. So the impulse starts from the spinal cord. So in a certain way, it's going to take it has to do this long direction from the spinal cord to the tip of the penis, to the, let's say, to the transplant of the foreskin over the penis.
Question number 2. I'm curious about the surgery itself. Some of us only had certain parts removed, like the frenulum. How would that affect the procedure? Would this newer theoretical approach still work for me?
Theoretically, yes. A patient could be treated who's just missing the frenulum, but we aren't at the clinical stage yet. But just doing something like the frenulum would be a specific question for the surgeon. So maybe it would be possible to regenerate just a frenulum and attach it surgically.
Question number 3. In the current research on tissue recellularization, does the tissue being recellularized include the frenulum in its original form or not?
Yeah, that's why we have requested to the protocol of cutting to save even the frenulum from the harvesting of the foreskin from the cadavers, because we want to involve even the frenulum to the transplant of the foreskin. So we should have even the frenulum to answer to your question.
If the entire tissue comes back intact, is it possible to remove the frenulum separately and transplant it somehow?
Well, as I said many times, the frenulum topic is a very detailed and specific project, because the frenulum is, sometimes 1 millimeter thick, and it's attached to the glands and to most of the part of the rigid band and the top part of the foreskin. So this, I would leave this specific technical surgery to the scientist, to the surgeon that is going to perform, because we need microsurgeon to attach only the frenulum. And as I said other times, we are considering to have the entire number of nerve endings even without the frenulum, because we will consider such a result acceptable. Because if we know that there are 100, I make an example, 100 nerve endings, okay, with the frenulum, and we can put these 100 nerve endings without attaching a difficult part like the frenulum, then to me it's fine. I think we have achieved the goal. But this is my idea. Just I have to talk with the surgeons about this. Maybe they have different ideas. Maybe they have already performed something so specific and so small on other patients.
Yes, if we can recellularize the entire foreskin, we could definitely recellularize a small piece of it, excise that piece, and implant that into a patient. But what Enzo was really getting at, like the meat of the question is how skilled is the surgeon? Can the surgeon accomplish the final surgery? It's going to be up to the skills of the surgeon whether they can reattach it. And so I like right now I don't see an issue with it, but again that's really a question for the surgeon at the end of the day. I would like to kind of add to that, and something that we only touched on really is that Dr. Ziron's team is very expert in this field of regeneration and urological regeneration. So prior to meeting, and one of the things that got us working with them is, so I don't know if we told you everyone the story of how we got working with them. And just real quick, Enzo found this group in Bratislava, and he was really looking at them for tissue donor collection. So we were originally purchasing tissues from this group, and we really helped them set up like how they were going to like sell tissues. This is how we transport them, stuff like that. So we kind of grew with them in that regard. And in the process of working with that and understanding that, he's a very skilled urological surgeon, he's really passionate about regenerative medicine. We found out that his own research is in the animal trial phase for urethral regeneration. So they're regrowing urethra and implanting that into, I believe it was rabbits at the time. I don't know where their personal research is now, but just to give everyone an idea, the team we're working with is very specific and specialized in penile regeneration. So they were working with urethra. They were working with, they were already doing foreskin collection off donors, off of cadavers for their research. So this is kind of a seamless move for us to find a team that was already doing something in the realm of where we're trying to do research. And when we have hurdles like this, when we say like, hey, can we get this like it's not like a long discussion trying to figure it out. It's usually a pretty quick, like, yeah, we can do this and this down the line at the clinical stage. Or when we talk to this surgeon, he's a microsurgeon. Let me talk to him because he has the expertise. So we definitely hear like your concerns, and we'll definitely bring this up with them. And then as time goes on, when we get to the clinical phase and we find the surgeons, we'll make sure that that's a topic that gets brought up with them.
Question number 4. Is there anything we supporters can do to help move this process along faster? Or is it really just a matter of time at this point?
You had said what can the donors do to help this project move along quicker? Well, one thing that came to my mind is we're always looking for opportunities to increase our outreach. Of course, we really appreciate it when people spread the word on social media and word of mouth and tell if they find other people who are open-minded on this issue and are looking for ways to help, to let them know about Foregen. Also, at the same time, we know, the media landscape out there is changing and becoming more decentralized. So if people are aware of different, podcasts or news outlets or public figures who may take an interest in this topic, we're always looking for referrals. We'd love to be put in touch with people who have a platform who may be interested in discussing our project or having us on for a talk to give more exposure to some of their followers. So if you are aware, if you have recommendations to us for podcasts we should look at, or not just that, whatever you want, not newspapers, TV shows, whatever, social media figures, please let us know. I think that there's always room for potential volunteer help from donors. For example, we're always looking for new moderators for the Discord, or even there's an unofficial subreddit that I think could use some moderation help. And furthermore, when it comes to social media, I know we talked about that before, but we could use potentially help with, I guess you would call it like a brand ambassador, or, for example, TikTok is a platform we've looked at in the past that's very popular, but we haven't been very active there. We stick mostly to Twitter and X and Facebook and Instagram. But if there's anyone out there with experience with social media engagement and publicity and outreach, we'd love to hear from you.
Question number 5. For the human clinical trials, will you be looking for participants who are partly restored or fully restored in order to gather a broader sample?
No, that makes perfect sense. To answer that question in short, we don't really know at this time because we haven't gotten to CRO yet. So once we find a contract research organization that would be helping us with our clinical trial package, once we get to that phase, so once we get closer to the clinical trial phase, we could definitely answer that more thoroughly. But to answer your question right now, we really don't have an answer because we're still vetting out the research side of it, the regeneration side.
Question number 6. Will each patient need a histological analysis of the recellularized foreskin before implantation? And if so, does that add months to the surgery timeline, or will it be faster once the technique is fully optimized?
I think the histological analysis of that is going to be performed now during the preclinical research phase, but also during the human trials. I believe during the human trials, but I don't think that would be a necessary component of the surgery when it's offered therapeutically for people in the future. So I don't think that would add any time to getting the surgery as a patient.
Yeah, so even if we did make that like a QC component of before every patient receives the sample, we take a small piece of the edge, so I mentioned the bioreactor growth method. If like, let's say, for example, let's say we overgrow the sample a little bit based on your needs. We take a little bit of that overgrowth, we send that out for histological analysis. That would be more like clinical, so that might be like that day turnaround. And you would get the call that things are ready once we knew that the sample was ready. We're not sure how that's going to look right now. That all depends on the governing body, how they want that done. So right now the plan is like we're going to do that as a QC step to make sure that what we say we're doing is actually happening. Did we recellularize it? Well, the only way to know is to go look at the cells. So that's why we're doing that in the research step right now. When we get to the clinical step, if like a EU governing body, for example, says, well, yeah, you have to do that every time, then we would do that in the clinical setting. So kind of like if you go to the hospital, like if you go get blood work done, or if you go get like a tissue sample processed, a lot of times that can be like a 1-day turnaround. That shouldn't be like a 1-month turnaround. Like I would say within that week of sampling, there should be answers whether or not that tissue is ready to go.
Question number 7. My understanding is that major blood vessels start forming in the bioreactor while the tissue is incubating. Is that right?
Yeah, so the vascularity is that's going to be something that we're looking at in that histology. So that is the goal. That is the goal for the major blood vessels to be formed and the micro blood vessels to be formed after implantation.
If so, how would the surgeon handle those vessels? Do they need to be cut and reattached, or do they just integrate on their own?
Yes to both. So some will integrate on their own, but we've talked a lot about microsurgery, like I think every call. That's maybe that's a really good question because that's a point that we could have been clearer on. So the microsurgeon, one of the big deals with a microsurgeon is that they'll know which ones they need to connect and which ones can kind of go on their own, to like minimize scarring and stuff like that. and then to maximize integration of that sample. So like the acceptance of your body and that sample together. So yes to both. Like the surgeon, we would have to have a microsurgeon that's skilled in that, and then some would go on their own. And then, to be honest with you, probably up to like a year after, you would see maybe some new blood vessels, maybe some new vein growth. Like I wouldn't be surprised if that's the case.
There's also talk about reinnervation. Could the major nerves form in the incubator as well?
The nerve is something that's the nerves are already in your body. So if if there's still living nerves in the skin that's already there that we're attaching to, enticing those nerves to continue growing and starting new nerve endings. And then if there's no nerves there, and trying to entice your body to stem a new nerve into that location. So the nerves are the harder part. We can't really just transplant um, nerve cells there and then expect them to grow. So think of nerves. I think there was a question posted to Enzo about like the nerve starting in the spine. Think of like the wiring in your house, right? Nerves kind of act like the wiring in your house. That if you want a new outlet, you can't just stick a new outlet in the wall and hope it, connects somehow. You have to have an electrician wire in from your main box, from your main power input. And that's how the nerves in your body work, that they have to stem from the main junction, which is your spine. So it's a little bit, that's why that topic is so much harder. And that's why we're working with Dr. Vega to try to figure that out.
Or is that aspect still uncertain?
Yeah, we have some good ideas. Like that's why I said like the cytokines and the growth factors to try to make your body grow that stuff once your body has accepted the new sample. But yeah, as of right now, we have some really good ideas, but we haven't gotten to clinical to try them out yet. So
I'm asking because in sexual reassignment surgery, they often use a nerve graft to move sensation further down. But here the distance is shorter, so I'm hoping the nerves could just grow in place. Does that make sense?
kind of. So in the case of reassignment surgery, they're moving an existing living nerve and they're reattaching it. So that's like in your house, like if you want to move an outlet, the wire's already there. They can move it to the other side of the room. They're just dealing with the wires that are in that room, right? But if you have no wires to begin with, you have to put that infrastructure in. And again, in the case of nerves, that all stems from the source. So it's a little more involved in our case. Um, yeah, I know that really doesn't answer your question. It just makes it more complicated. But the short answer is we don't really have a solid answer at this time. That's why we're working with Dr. Vega and the team at Bratislava. And that is definitely one of our big concerns, that once we get to clinical trial, we can actually start looking at that and implementing some of the ideas from Dr. Vega.
Question number 8. If I wanted to remove my old scar tissue, would that be done in the same surgery or in a follow-up procedure after Forgen's surgery?
That would be a question for the surgical team that is part of the clinical trial when we actually get to the clinical trial side. There's so the scar tissue, um, there's actually there might be other methods to remove scar tissue. So it might not be beneficial to like remove that tissue. It might be better to make an incision there to try to reattach the new sample. But that's all. That might be also a case by case basis. Like my situation might be drastically different than your situation. So from patient to patient, that's probably going to be a separate decision between the patient and the surgical team, like how they want to do that.
So we probably don't have much time left. Let's see. There's one more question in the chat. He says, maybe it's way too early for this, but he's wondering what public release might look like. Obviously, only a couple of surgeons will be providing the surgery for a few years, and there's only so many surgeries they can perform per annum. So yeah, it probably is too early to give a specific answer, but maybe we can provide a broad, general answer of how we anticipate public release to take place and how we could start those early stages of scaling up. Maybe, William, maybe you have some thoughts on that?
No, I think you answered that pretty succinctly. We really don't know till we get closer through this. I will say like you'll kind of get a idea of what that painting is going to look like once we get to. So it's not going to be like once we get into clinical trials, then we'll find out. It probably once we like start looking for a CRO. So the bar graphs that Ryan showed of like our current progress, as those start reaching higher, as we get closer to finding the CRO, we'll as we find a CRO, we're going to know how that's going to look, because that's going to be the team that's going to be doing the surgery, and that's going to be where we like obviously if we do the if we do the clinical trial in like Bratislava, then that would be like a big like, okay, how many patients can you accommodate in this amount of time, and we'll know that information more succinctly at that point. So not like I'm not saying like wait like 2 years, but like maybe I think like toward the end of this year into next year, once we start looking for a CRO, we'll have that answer. Like, what is the landscape going to look like? What is that actually going to look like as far as like being a patient and getting this done? We'll kind of have a clear picture at that point. If that makes, I know that doesn't really answer your question. It just says like we'll have a better answer at that time, but that's I think our closest to an answer right now.
All right, great. Thank you, William, for adding some specifics there. So it looks like that's a wrap. We've answered all the questions. Thank you guys so much for your thoughtful questions. Those are really good. Enzo and William, thank you so much for taking the time. And thank you all for all the donors out there for your generous support. You're really helping us move this project forward. Do you have any final thoughts, any parting thoughts you'd like to share, Enzo or William?
No, I just want to thank you all the donors, because they are really close to us. I feel, that they are participating to this activity emotionally, and this is what I like. So I'm very happy to work with these guys.
All right. Well, with that, thank you all for taking the time to speak with us. We really appreciate your support, and you ask fantastic questions. We're always available on Discord. You can reach me. You can email Enzo or William. And thank you for your patience. Thank you for support. And we are doing everything we can to get this project finished as soon as possible. So, again, thank you all so much. Have a great week, and we'll talk to you again soon.
Thank you, guys. Bye-bye.
If you would like to support our historic mission to develop a regenerative medicine solution for circumcised men, please click on the donate link in the description below. Your support is greatly appreciated.